But today we are going to hear from Dr. James Tucker. That's the abstract, not your biography. There we go. Dr. James Tucker was a research scientist, is now a research biologist at Agriculture and Agri-Foods Canada, just over on Grand Valley, and works with Anna that we had here a few weeks ago, although that was when I was sick, so I didn't get to see hers. But it's kind of a Barley Part 2 a little bit that you're going to hear today. He's got a PhD in plant science from University of Manitoba, MSc in quantitative genetics from Concordia, and a BSc in chemistry and biology from University of Winnipeg. And he's the current chair of the disease evaluation team of Prairie Recommending Committee for Oat and Barley. And he is also a member of the Canadian Phytopathological Society and an associate editor of the British Society of Plant Pathology's journal, Plant Pathology. And he's the principal investigator for a national barley cluster project that's been going on since 2018, which focuses on development of disease resistance in barley, which is maybe what you're going to talk about a little bit today. Okay, so I'll let you take over, and then when you're done, we'll have questions.
Okay, great. Well, thanks for the opportunity to let me come and present and come across the bridge and meet some people at the university. And I want to continue to build and foster relationships here. And so before I get into things, I want to gratefully acknowledge the research was conducted on ancestral lands of Dakota, Anishinaabe, Ojibwe, Cree, Dene, and Métis people. And so this is where I get to work every day. It's a beautiful place to come to work in the morning and hear birds chirping, and I'm grateful that I get to do my research on these lands. So Anna last seminar or 2 seminars ago talked a bit about barley. So assuming everybody's been given an update on what is barley, it's one of the primary crops grown in the country here. And so I'm not going to spend too much introducing barley, but on the other hand, I'm going to introduce some of the problems that are associated with growing barley, in specifics to growing barley and combating some of the disease problems that exist. So I'm a member of a committee that meets once a year, and we evaluate all the lines that are being presented by the public and private sector and so this is to help give a recommendation to CFIA to register a variety to be grown in Canada. It requires that we make this recommendation, CFIA is ultimately responsible for the allowance for the variety registration, but we as a committee talk about some of the merits, and I'm on specifically a disease evaluation team. And in that, there's a lot of diseases in barley. Barley's challenging. It's got a lot of pathogens and which cause economic harm to barley. So I've just listed up here. So these are all the diseases that we work on, and so these get evaluated on all the lines that are being put forward to be registered to be grown in Canada. So I'm going to focus my talk on some of these things to give people an understanding of some of the problems that we have. So these are just the diseases that are considered to cause economic harm. So there's a lot of other diseases that exist out there, but these are the ones that generally, under high enough disease pressure, will cause economic harm to farmers. So in Canada, we take a bit of a approach to protect the farmers in establishing merit and criteria for what is acceptable to register a variety. So you can see here, it's listed as a priority 1. So we used to have other priorities in the committee and we felt it was a bit confusing. So we've eliminated them from this table, but this is basically now all varieties need to achieve a minimum of intermediate level of disease resistance. And some of these diseases like Fusarium head blight, you can see here, it's listed as a 4 for ease of breeding. So this is quite a difficult disease to breed for. So I'm going to talk a bit about that in later in the talk. So first of all, I'll talk about stem rust. The stem rust, Puccinia graminis. So this is a picture from my nursery, and you can see this, the rust-colored lesions on the stem is an explosion of spores. So what happens is a spore lands on the surface of the stem and enters the stomata. And from that point, it silently invades the plant, and it grows cryptically without the plant really knowing that it's there. And it goes through its life cycle as a biotrophic organism. So it is specifically designed to infect the plant without causing damage to it initially, but eventually the stems just fill up with mycelia and cut off the nutrients to supply to the seed. The stems get weak and they break, and basically the seed falls on the ground. So it can be quite a devastating disease. Here's some of the foliar diseases that we're dealing with. So the first one on top here is spot blotch. So this is caused by Cochliobolis sativus. On the bottom here is Rynchosporium commune. So this causes scald. So you can see this water-soaked bluish lesion. Here's a symptom of scald. Spot blotch are these leopard-type spots. And so these pathogens usually come from the residues that are left in the field after the farmers exited the field, the bits of the stems and seeds and things that are left there. The pathogen overwinters, either that or in the soil, and in the spring, so that these spores are splashed with rain onto the leaves, and they work their way up the plant, and eventually they end up on the flag leaf. So what I'm trying to represent here is the progression of spores. And once it gets to the flag leaf, that there's a lot of photosynthesis that is involved at the flag leaf level. So that's really causing problems in the nutrients that end up in supplying the development of the seeds. So the spot or the pathogens, the foliar pathogens, are pretty simplistic in the sense that they basically function to reduce the photosynthesis of the plant, and you end up with thin seeds or death of plants. So another pathogen we work with are the barley, the covered smuts. So these are surface-borne, so they're found on the seed coat. There's genetic resistance that is generally available, and it also can be solved with seed coat treatments. So however, we do assess this, and we do require a certain level of resistance to the smuts. This can be particularly problematic in organic systems where they can't use seed dressings and the seed, these the smut bodies, basically there's just chunks of spores, and these things end up being harvested with the seed and end up on the seed coat and reinfect what ends up in the next year's planting. So, and this here is Fusarium head blight. So I mentioned earlier, this is like one of the most difficult diseases that we're dealing with, Fusarium graminearum. So it produces a mycotoxin in the grain called deoxynivalenol. You can see this picture here. This is a seed that I've sampled at a farmer's fields, and that looked something like that. So we plated it onto PD, potato dextrose agar, just to identify the species. So it's growing off. So Fusarium's a little difficult to understand. When you go to the field, this is a picture out of my nursery. So there's a lot of there's a lot of infection, but it doesn't take a lot of infection to ruin a farmer's harvest. So you might see like the odd kernel here and there, and it's also very hard to tell in barley. It's not totally obvious. Obviously these seeds that are really thin and stained are infected, but a seed that's infected later, it might retain its plumpness and it might look like a healthy seed when this matures. So it's a little hard to tell, but so the industry uses DON or vomitoxin or deoxynivalenol as a biomarker for this. So there's a lot of scrutiny and testing. This says the life cycle I described before, where the spore residues end up shooting ascospores for the sexual stage. These land on the heads. And so the Fusarium graminearum has a monocyclic lifestyle. So it has really only, it can produce secondary spores through infection. At this point, it can produce asexual conidia, which will be splashed around, but primarily it comes from like a main infection of ascospores that are wind-blown. So these spores are shot up into the air, but with a extreme trajectory and pressure, and they'll shoot up like over a meter over the crop and get caught up in the wind and blows around and lands on other plants in the field or fields away from this. And so that from that point, it produces, there's mycotoxin produced in the harvested grains. So this is a cartoon I produced of ribosome. So there's probably people here that know more about ribosomes and the chemistry. However, just simplistically, what happens is the DNA is encoded into mRNA, and that's brought to the ribosome, and the tRNA is used to create protein chains. So in reading this and producing the different amino acids in the proteins, the deoxynivalenol or the vomitoxin, it's a general protein synthesis inhibitor. So it can harm anything that have ribosomes and that create proteins. So it's basically a problem for everything. And so as we have ribosomes, and we produce proteins. Deoxynivalenol can be a human health factor. It also affects animals that are fed grains contaminated with this, because what happens is this DON gets caught in the channel that the mRNA is moving through, and it interferes with protein synthesis. So it's a general problem, and so it's highly regulated. So malting industry has a very strict standard of grains. So in the malting industry, there's a 0.5 part per million level of tolerance, which is basically at the level of detection. So if you find DON in malt, it ends up getting rejected. And so what happens to a farmer who's looking to get their premium for sale of grains into the malting industry, they lose out on that premium, and then they have to sell it into a less lucrative industry, such as the animal feed industry. And at certain levels, say for hogs, it's also critical to have a limit on the deoxynivalenol. Certain animals like swine are more susceptible to it, and cattle can handle it a bit more, but they also have a limit of 5 part per million. So once grains reach that level, they become basically unusable. So I'm going to talk a little bit about history and evolution. And Anna talked last time a bit about barley and the fact that it's an ancient grain. It was domesticated about 10,000 years ago and then brought from the Fertile Crescent. And then right here is brought up into Europe by Neolithic farmers. And so there's a long history, 10,000 years or so of people using barley as a food substance. And so there's over that time, the pathogens develop relationships with the host. And there's certain pathogens like the scald pathogen, Rynchosporium commune, they quickly became associated with barley shortly after domestication. So there's a real long history of coevolution between the host and pathogen, whereas in Fusarium graminearum, this may have occurred more recently in history. And I've seen it described in the paper that I've referenced here. They described it as an endophyte that's a situation that's gone bad. So they are still going through evolutionary pressures. I'm going to talk a little bit about the gene for gene hypothesis of plant pathology. I sort of assume that here not that many people work that strongly in plants. So talking about plant pathology, which is a subdiscipline of botany a little bit. So there's a crediting this man here, Henry Floor, who's credited for the gene for gene hypothesis. So that which states that pathogenicity genes have a one-to-one corresponding relationship with the host resistance genes. So the host and pathogen evolve together to a point of equilibrium where the host has a resistant gene. If it finds the corresponding avirulence gene, there's a lock and key mechanism where the host recognizes the pathogen is there, and it triggers a hypersensitive resistant response. So in all other scenarios where the host doesn't have the resistant gene, or the host isn't recognizing the pathogen's avirulence allele, then there isn't resistance. So just to talk about a little bit about the zigzag model of evolution of plant immunity. So this is a graph where the amplitude of defense is on the y-axis. So the plants can't move away from danger. They're not mobile, so they've developed immune systems to deal with the fact that they can't run away from potential hazards. And so the plants have receptors that called the PAMPs, or pathogen associated molecular molecules that are associated with pathogens, so say such as chitin or flagella or other things. And so the plant will has a receptor that recognizes when these things are present, and it triggers a PTI, or pathogen triggered immunity. And so what the pathogen does is it evolves effectors that helps combat the level of resistance that the plant is imposing on it. And it with effectors, so these effectors could be any sort of chemicals that the pathogen is putting into the host, like certain things like enzymes to degrade cell walls to gain entry, or anything that gives it an advantage to defeat the resistance, and this called effector triggered susceptibility. So what the plants then do, they have in the cytoplasm, they have molecules that are floating around, and they're looking for these effectors. So once the lock and key mechanism happens, these molecules recognize that the effector's in there, and so then it triggers a hypersensitive response. So it'll trigger an extreme reaction, so that to kill off the tissue around the pathogen, or also send out an alarm bell throughout the plant to increase its level of resistance factors. The pathogen then evolves new effectors to combat that, and then the host then develops a new resistance gene. So it's a back and forth play between pathogen and the host. So through coevolution, they reach a state of equilibrium. And so once these genes exist, pathogens and plants will cycle through under selection pressures. The pathogens will be selected for this effector or this effector. So depending on whatever resistance gene has been released in the variety, it'll impose a heavy selection pressure on the pathogen to move towards one or the other way. So that is talking about major gene resistance. So I'm going to give some examples about major gene resistance, some of the work we're doing at the research center on this. So this is a picture of Robigus, which was one of the gods of the Romans. So stem rust's been around for a long time, and in April they would have sacrificial festivals in hopes for a good year without rust. So this goes to show like how long the rust pathogen's been around and affecting cereal crops. And so at the research center, we're working on RPG1 stem rust resistance. And so this has protected barley for nearly 70 years. So there's just this one single gene that confers resistance in barley crop. It's been highly effective. This is a really bad strategy to rely on a single gene. As you can see, what I was talking about before, the pathogens under high selection pressure to move towards overcoming this resistance. Somehow it hasn't happened in a major way yet. So it has happened though. So in domestic and foreign environments, there's races of stem rust that have been found. So in North America, in the late 80s, early 90s, there was a big worry about the QCC JB race, which was able to defeat the RPG1 resistance, and also more recently in Uganda in 1998, they found a virulent race rust, which was termed UG99, also known as TTKSK. This is evolved from that point. So initially it was found in Uganda, and it's spread all throughout Africa, and it's evolved different forms of virulence. It's spread into the Middle East, and there is a concern, if this spreads into areas such as Pakistan or other areas of the world that are heavy on producing cereals, it could cause mass famines. More close to us in recent time, there's been virulent races found in Washington and US. So we're working with molecular marker for RPG4 or RPG5. It's a gene complex. So this confers resistance to the QCCJ and the UG99 virulence races. So we have molecular markers that are used to for breeding on that, and maybe Dr. Badia had mentioned this. We recently had a variety that was supported for registration that has rust resistance in this form. So this is a picture of our rust nursery at Brandon. So I don't spread the virulent African race in here, but we use the QCCJ race as a proxy for screening for the UG99 race. And so what we do is screen lines that are positive for the molecular marker, and these are planted into the nursery. And so these are spreader rows, and these are spray or needle injected with spore from the stem rust, and then it quasi-naturally blows into research plots, which you can see here. They're like a clump of 10 seeds that following the infection, I go through and rate the plots for the percentage of the stems that are covered and the type of reaction. So part of it is for breeding and for Dr. Badia's program and screening her lines for resistance. One of the exciting things that I work on is trying to find new genes or new genetic materials. So this is just a graph of some of the lines that we have been identifying. So some of these, this is our check line, or where the original donor of the RPG4, RPG5 gene came from. So some of these lines been screening from Plant Gene Resources Canada. They have a certain level of resistance that's comparable to that. And so the hope is to find some of these lines that potentially carry new genes that we can incorporate into the breeding program. This is a bird's eye view of the nurseries at Brandon. So this is the spot blotch nursery. This is the stem rust nursery. Putting this up here. So Stephen Holden did his master's work here. And so for several weeks, he was flying a drone over here and collecting data on spot blotch. And so he also harvested this area. So you can think of like this is a full-size pickup. So I think he harvested about 30 pickups worth of barley. So as I just give Stephen a shout out so people know the level of work that was involved in this. And hopefully he can get some really good predictions from this. The work So I'm not going to talk, even though I work a lot on spot blotch, just the methodology, like we just plant clumps of seeds in these large nurseries and then screen it and score it on a 1 to 9 scale. The research we do is very applied, and it contributes not so much to academia, but into products. So this is AAC Synergy, which is the new king of barley. So you see any of the AAC lines or AC lines or Ag Canada lines. So you can see Synergy 2023 is the dominant malting barley in Canada. So this was selected, I think, in probably it's about 25 years ago now, and out of the leaf disease nursery, one of those little teeny plots, somebody clipped 10 heads from that, and now it's like grown at probably 3/4 million acres of land. So it's really to show how long it takes to produce a variety, but it's also these little things end up in big things. So the research is this is the deliverable on the research. It's we use scientific method, but in the end we are working for Canadian farmers. And one of the accolades of Synergy, beyond its superior malting quality and yield, is the fact that it has a very good resistance to spot blotch. So it was selected out of our leaf disease nursery, and it is now #1 in Canada. We'll talk a bit here about the difference between qualitative and quantitative traits. So up to now, I've been focusing on qualitative. So these are major gene resistances. So like I said, you have that resistance gene in the plant. It finds the pathogen through its immune system recognition and triggers an extreme response. So you see very different phenotypes. So something with a resistance gene is very little, probably like a little speck of something that's on a leaf, whereas if it doesn't have the resistance gene, it's a completely different story. When you're dealing with quantitative resistance, these are shades of gray. They're very hard to understand. They're controlled by multiple genes, so it's not the low-hanging fruit of research. It takes a long time to develop resistance. However, it's known to be very durable if it's developed. So it has some advantage. It just takes a long time getting to that point. So quantitative resistance is very challenging and confusing. And the fact that so when you have so it's basically controlled by many genes of small effect. So in each of these genes have an significant environmental component. So it's a very different beast than dealing with major gene resistance. And so a lot of people haven't been able to make progress in it because it takes so many resources. It takes a lot of labor, and to assess these things, you need large populations. And I'm mentioning this because quantitative genetics is the basis of our Fusarium head blight resistance. We grow a large nursery at Brandon of 14,000 plots, and it's of these plots we harvest approximately 10,000 of them by hand. So this is a major undertaking, and so the nursery at Brandon is the main hub for doing this in Western Canada. So we have backup locations in Ottawa and Charlottetown and Morden that I send the most advanced materials, but we do most of our work in our nursery at Brandon. And you can see from this pie chart, most of it is to service the Western Canadian plant breeders. So I service so Dr. Badia's program, and also Dr. Aaron Beatty, and Dr. Yadeta Cabeta. So Aaron Beatty's at Crop Development Center, and Yadeta's at Field Crop Development Center at Olds College. And so they're sending their breeding lines. We test them, infect them, and then we do the mycotoxin analysis through ELISA chemistry. And so we're producing over 10,000 data points a year. So that's the primary focus. Also, along is population development. So in working with Dr. Badia of segregating populations into the nursery, and I walk through and make selections based on things that have a level of resistance in agronomics. And so we test them for Dawn, and then they get advanced in her breeding program. So we put F6 generation. So once it's the lines are fixed or becoming fixed, so that we can actually make an assessment on them. And then she, from that point, puts them into progeny rows, and then they get advanced through the breeding process and looking in yield plots. And so that's kind of the service work that I contribute to. One of the passions research that I have is looking at germplasm introduction. So these would be international or other reputed sources of resistance. Also just screening for new resistant sources. So in Canada, there's been a rapid shift of the population of Fusarium graminearum. So it's shifted from pathogens that have that are defined as a 15 ADON chemotype, and they're being replaced with a 3 ADON chemotype. We did some research with Randy Clear at the Grain Commission, where we spread our corn on the nursery in a certain ratio of 50-50 of each chemotype. And then we recaptured grains and then did a single spore isolation from the grains, and found in certain years, certain chemotypes dominate over the others. But he also found that the 3A DON chemotype generally produces a higher level of DON. So with the same level of infection, you end up with more toxin in the grain, which is not a great situation. So that's the main pathogen, I guess, in the prairies now. I'm glad Sydney's here today. So you can see, I don't think she's smiling very much here in this photo, but so this is just an idea of what it's like in our nursery at harvest. So we harvest 10,000 rows like this. So we're hand sickling with rice sickles into these Rubbermaid tubs. And this is a better view to perspective of the amount of work that's involved. So I'm out there, I'm working the combine here. So this is a group effort, and it's a massive effort. We lay out harvest bags in the plots, and then each of these plots are sickled into a tub and brought to the combine and threshed, and then the grains are kept in the bag. And then people spend the winter cleaning the grains and grinding them into flour, and then we take a subsample of that and we send it to several chemistry labs to get a measure of the DON. And all the breeders want their results like immediately. So we use 4 different labs so that we can get results back to them, and hopefully we'll start getting results back in the next month. So we got to keep the breeders in data so they can make their decisions, because breeding is all about not what you put forward, but what you throw away, because every line that goes forward costs resources. So I mentioned one of the passions I have is working on resistant sources. And so this is just a diagram from Plant Gene Resources Canada. So they located in Saskatoon, and we are very lucky in Canada to have like the best germplasm gene bank for barley. So you can see there's over 40,000 different lines that they have in storage. And so this is a really valuable resource. So this comes from missions that people, went and traveled the globe in the 70s and collected land races from all sorts of countries, and farmers in different countries that would save seed, and they would select and basically do their own breeding to select for their regions. So this is what they look like. This is some of the typical, what you see is in some of these lines is they're very tall, they're very weak. So this isn't even at a point where the grains are filling the head and they're already starting to lodge. They've extreme maturities, so they'll either head out very early or very, very late. So there's a lot of problems in working with this kind of material. So over the years, so last 5 years, I've been looking at 500 lines per year, walk through them, select them for their level of disease in the heads, and select them for something that has a certain level of agronomics that a plant breeder might use. Because if it's too far away, it's like turns into the 30-year breeding project. So it really taking too many back cross. So we want to have something that's a bit closer to modern cultivars, but yet has lower mycotoxin. So this has been effective. So AAC Goldman and Low are commercial varieties that are pretty much the best varieties that we do have for Fusarium resistance. And you can see some, quite a few of the lines that we've selected over the years have lower mycotoxins levels than these things. So they represent foundations that the plant breeders can use for crossing. And Dr. Body has made several crosses with these lines. So it's maybe, when I'm on my deathbed, I can look back and there'll be one of these lines that's somehow moved forward, because it will take a long time to incorporate. I didn't want to bore people too much about all the lines that we look at, but some of these specialty projects that we're working on, and this is one on heritage barleys. So these are lines that from the previous century. So there's still there's sort of a revived interest, especially with the all malt or craft malt industry, because these things are reputed to have amazing beer flavors. So people are starting to bring them back and brew with them. And so you can see this line here, Chevalier. So this one, I guess, was at one point the number one variety in England. So these lines, they've been exposed to an environment. So Fusarium head blight came from Europe to Canada. It's so these lines have been under some selection pressure for a long time. So the hope is they could be used for flavors and beers, but they're also potential for use in Fusarium resistance. And so I looked at originally working with collaborators in Charlottetown with biologist Dan McEachran and scientist Aaron Mills. So they were looking at these for agronomics and some of those beer flavors. And in Eastern Canada, their environment's a lot more like what you would have in Europe, with more moisture in the growing season. So they were testing them out. And then Dan put them in his nursery, and he had some initial results. And so I asked him to give me his best lines out of the Charlottetown Fusarium nursery, and I grew them in Brandon. And so this is the deoxynivalenol content of the grains over 5 years. So as mentioned before, AAC Goldman is our standard for breeding in the program. So was derived from an exotic Chinese cultivar. So it has a very good level of Fusarium resistance. And you can see a lot of these lines have a fairly high level of dawn resistance. So working with Dr. Badia, we did grew them in yield plots, and we did malting analysis on them. And so some of these lines, actually, have an okay level of yield compared to modern cultivars, and they do have a certain level of malting quality. Generally, they didn't have like malting quality to some of our current varieties, but there are better ones in the group, and so suggestions have been made to her for breeding purpose. So it's hoped that we can bring back some of the genes of the past and to put them into the current malting cultivars. So this is a little bit about what somebody can do with information like this. So that generally was as a smaller group, but somebody can do association mapping. And this is association mapping of not of the exotics, but this is a panel of 400 Canadian barleys. This was tested over 3 locations over 3 years. And so this is a principal component analysis of the different groups. So you can see the Ag Canada group sticks together, and same as the Crop Development Center and certain other groups, they show clustering. So when you do association mapping, you need to account for that so that you don't end up with false positives. This group was genotyped by Dr. Colin Hebert at Ag Canada at Morden using a 50K SNP chip. So this is, so across the whole genome, there's 44,000 single nucleotide polymorphic markers. So this is the kind of data that you can use to handle quantitative genetics and looking at variants and try to make associations over the whole genome. So you can see there are along this axis are different chromosomes. So each color represents a different chromosome, and any of the dots above the line indicates a marker that's statistically associated with Fusarium head blight or deoxynivalenol content. And so this is published in Canadian Journal of Plant Pathology. Unfortunately, in Fusarium, we didn't find any major genes. So the hope of this would be to take one of these molecular markers, and then if it was contributing 20% to the phenotype, we could breed with it. All these markers, they're symptomatic of quantitative traits, and they're contributing maybe 5% to the phenotype. So it was a learning lesson of the problems associated with Fusarium head blight in barley. It's even more difficult than in wheat, where they do have major QTLs. So we do have some markers, and that maybe can be used in a genomic selection program. So speaking of that, this data was also used for genomic prediction. So here, this is just an example where you take the population and you through ridge regression model or others, using that to make a prediction of the phenotype based on which it's different from association mapping, where you try to find in each individual marker that is important. In this situation with genomic prediction, you don't really care about individual markers. You just want the sum of all 44,000 genetic variants together. What do they contribute to the trait? Can you create a regression on the phenotype, and so you can identify a breeding value of each of the markers, apply this in a model to make predictions. So Generally, people do this. They take a training set and then they'll partition out a validation set. So use this part for modeling, and they try to predict this without knowing the phenotype, and just so it's just trying to run a correlation of how well you are predicting. This is from a paper here in Frontiers in Plant Science, and it's looking at different models. So this is the general accepted model, the RR-BLUP. So this is it's very good at predicting Fusarium head blight, and also decent at producing a modeling for dawn. We also working with a data scientist at University of Manitoba with trying to see if we could apply machine learning techniques to make better prediction. But the results from this study show that we can predict quantitative traits like Fusarium head blight with whole genome markers. So in this case, these are correlation values. So in R squared, probably around 0.4. So we can then predict at least about 40% the genetic variance. So it's enough to make a simple selection. So you could select on lines and not have to put them into the field. The whole purpose is like to limit how much of the field work we have to do, and making students with sad faces like Sydney. So when we have to harvest 10,000 lines, so this can be applied when plants are about that big and just a little sprout, and you can take the DNA and do a testing and then make selections. So we haven't used it so much for that purpose, but we're still validating things and using it as extra data, almost like, how we use like our external nurseries as extra data. We use it's just another form of data, but somebody strictly could just select just on this and improve populations, just by the markers. So I guess I'll probably end with just crediting some of my funding. So I'm funded through National Barley Cluster, also through a core agreement for commercial development, and working with Dr. Beatty at Saskatoon at Crop Development Center on phenotyping and doing genomic selection for dawn resistance, and also with Olds College for that. So a lot of funding for applied research to develop varieties with low Fusarium and dawn in them. So I say cheers and thank you for letting me talk.
Hi, thanks for coming to present. So the Fusarium head blight that's in Africa, that's the really devastating strain of barley, right?
Or the strain of Stemra.
G99 is the pathogen from Africa that's
so in Africa, the stem rust pathogen Puccinia graminis, it has virulent races. So what a race is a form of the pathogen that has a direct gene for gene relationship. So so there are these different races that are found, and they've evolved, and so they're identified basically through putting on wheat differentials. So they have because stem rust can affect wheat and barley, they use wheat to define the races. So they have these sets of wheat lines that have one gene in them, so they can tell if the race or the pathogen will knock out or overcome that one gene. So that way they have this complex system where they define a race by how virulent it is, basically as your alphabet goes up. So a T is pretty high up there. So this one is the one that's TT and then KST, or those are basically it's extremely virulent on a group of different differentials. So something with a lower alphabet would be it's like a weaker race. So these are pretty devastating pathogens.
So if one of those came over to Canada, one of the really virulent races, like how would we actually be prepared for something like that, or would it kind of just go across our country and
I guess the good thing about stem rust is it can be controlled by pesticides. So we could kill it with pesticides, but the whole point of genetic resistance is we're trying to work towards pesticide-free production of barley, because pesticides are quite expensive and they're environmental pollutants. But it could be a problem. And before they develop resistance in Canada, there's histories of really heavy stem rust epidemics where farmers, it's a really severe disease, and it cuts off all nutrients to the head, so there's basically no seed left at the end. And there's pictures of farmers trying to harvest their cereal crops, and there's these black clouds of spores, and so they would barely get any harvest. And that's one of Canada's global contributions is some of the cereal genetics that's been conducted in research here to develop some of these resistances. It could happen, and we hopefully Dr. Body has got this resistant variety. So we have a line on the shelf that even though it wasn't commercialized, it's there. So it could be put out and farmers could use it. It's a challenge when you're incorporating genetics like major gene resistance, because it requires energy, the plant. So some of these resistances in the past we've been incorporating, it's been a long time coming. So Dr. Body, his predecessor, Dr. Legg, worked on this in the 80s. They had massive nurseries, just like my Fusarium nursery, making selections and to develop. He never got quite to the point that she's at for yield. So she has produced something that is close to the yield of a modern cultivar, but has that higher level of resistance. So it's there, and it didn't get registered, but it's if there's a massive stem rust epidemic, it's probably be pretty quick to register it. So we are breeding for now and breeding for the future. So it's a lot of these tasks that we're doing. It's for what may happen, not necessarily what's happening right now. Sorry, that's quite a long answer for.
No, that answers it very well. Thank you.
Probably a very general question. I'm just curious how immunity of plants compares to that of animals.
Well, it's kind of similar in what's floating around in your blood is detectors, in the sense you have blood cells that are designed to recognize things. So once a triggering happens, once that lock and key mechanism happens in a plant, there's a bunch of hormonal things, cascades, things that happen. They tell the plant sends signals to reinforce lignin and things to stop pathogen penetration. There's a lot of there's an immediate response of things, so that the hypersensitive response basically just triggers reactive oxygenated species to kill that pathogen spore or whatever is infecting, and kill the whole area to stop its spread. But there's also a lot of hormones that signal other defensive mechanisms to just like if you're infected with something, your body says, let's increase our immune system. So you encounter cold, or you get infected by something, your body will produce defenses. The plants do that as well, but it might be even in a more severe situation where they'll just kill a complete area of the plant, sacrifice a bit of the plant so it doesn't spread. You can see people sneezing and you just walk away, whereas a plant can't really do that. It's got roots, so it's sort of stuck. So it's got a very extreme, I guess, defense mechanism. I'm not that familiar with human systems, but I've been sick before, so I've.
Any other questions? Oh, you go. Oh, sorry.
Very nice talk and informative. I was just wondering, how do you do the genetic parameter analysis on the crops?
Do the genetic
Parameter, like what are the genetic morphology, or if there is disease growing, if you find out through the this kind of biochemical parameters, so like in laboratory.
How do I recognize the disease, or how to?
No, the genetic combination that you showed in one slide. I don't know, in the middle, one of the slide, there was some kind of genetic information.
Okay, maybe I'll try to. You can tell me when to stop.
Sure.
Which keep going. Like is in.
Yeah. This is the one, genomic prediction. Genomic prediction. And the other, this one.
So these are done by like complicated statistical models. So it's basically like a fancy regression. So you take a giant matrix of all your marker states. So you'd have 44,000 markers, and for each of the genotypes, they either have absence or presence information. So it turns into a very lopsided equation where you have 400 or so lines on genotypes, but then you have 40,000 markers. So to do a regression on something like that, it needs fancier statistics, like a ridge regression model that allows for that kind of situation. So basically, that type of regression model will allow for these situations. And so basically it assigns a breeding value to each individual marker. So if the marker is like present or absent, if it's, say it's like a 0 or a 1, they say if it's a 1, then it gets that breeding value assigned to it. So it solves for the fact that the markers each have a value which is like a slope of that regression for each individual marker. You can imagine it gets fairly complicated. And so that breeding value is associated with that marker. So that breeding value is very small, but each one adds up a little sliver to positive or negative towards the trait. So each of those slivers add up. And so what you want in a resistant variety is more positive slivers than negative slivers. And the breeding value assigned to each marker is just a fraction. So it's complicated to do this. In this study, we also used different forms of machine learning approach, which basically abandons statistics. It's just a different way. And everybody's thinking that machine learning is going to solve everything. But in this situation, the statistical models were just as good as the machine learning, and that's maybe not, you know, certain situations, the machine learning may or may not be good at predicting things better or worse. So but it did, I was actually surprised that it did predict this well. So, and this is something that maybe it's a different way to look at things. It's not a statistical, but maybe it's finding patterns that don't necessarily follow the statistics. So it could be complex patterns, like in the statistical model here, it's just looking at individual markers and the slivers adding up. But maybe the genes that interact with each other are more complicated scenario, and maybe the machine learning could predict that scenario better. And that even though we weren't able to show that there was an improvement, it's a totally different approach than a statistical model. And so it could potentially be upscaled. And then this population was only so big, 400. But if you added 4,000, maybe that approach would do better. So it showed that it at least did as well as the statistical ridge regression.
Okay, thank you. Do you take any specific part of the crop as sample?
Sorry?
Any specific part, any particular part of the crop as sample, like on what is the sample from the crop to check these genetic expressions?
Which part of, like, for the DNA or?
Yeah.
So yeah, we just take a seed and germinate it and grow it into a little seedling, and then the leaf is cut. And so that's a good, it's, a highly growing part, so there's lots of DNA, good. And so that's DNA is taken from something that could be as like young as 4 days, and you could get a result compared to something that takes 4 months to see. We can't even do growth cabinet work for these things because it takes 4 months to grow a barley plant. So it takes a long time to wait. So we focus in the field, so something is the advantage of genomic prediction is that you can have a result very quickly. If you had the lab all set up and you could get the DNA, send it to the lab, they could run, they have to hybridize the DNA to these microbeads of oligo extensions, and so that takes a little bit of time, but you could have a result in a couple of weeks versus like 4 months. And so this is sold to the world as something that could improve the pace of genetics. So you could have, quicker cycles. So if you kill things, you don't necessarily have to keep a lot of them around. So you could progress your genetics quicker.
Do you still want to ask you a question, Pam? We're about 12 minutes on, but
Maybe just a quick one.
okay. So this will be the last question then.
Yeah, sorry. So yeah, we'll keep it quick, and if it's got a really long answer, we can just leave it. But is there work going on as to the role that climate change might play in disrupting these coevolutionary relationships that have evolved over very long periods of time?
Well, I'm not specifically doing research myself, but we are growing these things in the world, in the real world. So the environment is changing. So I'm not targeting like climate resistant genes, but my nursery in Brandon used to be a net blotch nursery. Now it's a spot blotch nursery because spot blotch is a warm loving pathogen. It has displaced the Pyrenophora teres pathogen, which I don't even see anymore. So in some ways we are working in the real world. I would like to do more research on climate, understandings, like, we have growth cabinets at work that we could, increase the temperature. We could increase CO2 levels. We can do all these things. We just, gets complicated, but that is like basically where the government of Canada is going. We are like looking forward to meet our goals, and my contribution to environmental goals is to produce plants that can resist the pathogens without spray, which means you don't have to drive a tractor across the field and burn diesel. So we are conscious, but I think we also need to move towards that, targeted research. And maybe in the next couple of years, I'll get into that, because some of these things like spot blotch is definitely seems to be getting worse. Some of these pathogens, and we don't know how they're going to react in climate. So, so we maybe need to test them, because they could favor the plants, or they could favor the pathogens. So we don't know.
Interesting. Thank you.
Okay, join me in thanking Dr. Tucker once more.